entities. So drug discovery process could be like, like finding a particular earlier days I'm talking of, finding a needle in a haystack. So this is what they were trying to do those days. So definitely that at some point of time, they will find out the molecule against that particular disease with the molecular marker or something, or the drug also can be developed after some years. But then it would be just like finding a needle in a haystack. Other than the drug discovery process, is these days has to be changed to a simpler, different type of scenario, and that is what I'm going to talk about these days. It's about simplifying disease complexity such that we can identify indispensable targets. By indispensable, I mean the real target which actually people should focus upon, instead of like thinking, okay, this is an elephant and I'm working on an eye, I'm working on the tail, and okay, this is, I found the tail. So no one thinks of the bigger picture. Earlier days, no one thought of this kind of bigger picture, and that's where the complexity lies, and so we need to really simplify. So my talk would outline these issues, and I'm working on this kind of multi-drug resistance and drug side effects. My perspectives of my vision, and my strategies I'll talk of, and how I've developed these kind of things, and what are the models. I'll discuss two models. If the time permits, maybe I'll just go for some funds and support acknowledgements. So the issues which are ongoing across the decades are nothing but this kind of different infectious diseases. If you see the rates of infection versus mortality, you will see that the bacteria has gotten us. Like they're very clever, and they are developing multi-drug resistance, and we are wreathing in pain. And there's some kind of antibiotic problem also. There's a vaccine resistance and antibiotic resistance also as well, or going on over the years. And the development timeline of these antibiotic classes have halted around about 2000, 1990s. So after 2000, there's hardly any such new discovery of new different classes of antibiotics which can help us. All those earlier ones, bacteria has got used to with it and has now developed multi-drug resistance. That's what it is all about. Or else, if you can just think of different idiopathic diseases, what is that? Idiopathic is diseases are those for which there's no such, like a particular cause being discovered. People are still struggling for cancer, for diabetes. Some people do talk about certain molecules to be targeted and all, but then still, why people are, like dreadful for the particular disease like cancer? So that means that people are still unable to actually tackle cancer. So there are over than 100 types of different diseases. Some of them can be autoimmune and so on. And even if you take drugs, for example, chemotherapies in cases of cancer can still be a dreadful picture for the sufferer, because once the chemotherapy is done, the normal tissues, the normal cells are also getting affected. So, or even forget about cancer, just normal any other disease, you take some drugs, and then there would be side effects. Okay, so why is this side effects? That is what actually we will be talking of. Okay, so, and these actually pertains to the kind of global issues wherein the Sustainable Development Goals talks about the good health and well-being. The third of them being, focused through my research, and I've been actually working on tackling multi-drug resistance and finding out certain biomolecular, markers against cancer, diabetes, rheumatoid arthritis, ankylosing spondylitis, schizophrenia, autism, and so on, just to name a few. So the perspectives of my research is that I really have a mission to simplify disease complexity, focusing on multi-drug resistance and side effects. I'm actually utilizing big data analysis, some interactive analysis of networks, and some structural bioinformatics. Okay. So these are the different techniques that I use: protein interaction network analysis, the single nucleotide polymorphism analysis, the network, the natural products metabolomics also. So then there could be some kind of virtual screening of this kind of bioactive compounds, and certain pharmacokinetic studies as well, which can help me to realize my vision. through these kind of strategies. So what I generally do is I retrieve the kind of proteins from the literature or the databases, find out a kind of, I build up a network, which could be maybe sometimes one people can think of as arbitrary. I generally prove them that these are not arbitrary, but the real networks. And I actually analyze them through centrality measures and core and modularity. I'll talk about these as well a little bit. And then I'll go for sometime a drug repurposing through some kind of virtual screening of pharmacokinetics, because sometimes some of the bioactive compounds are maybe proposed, or even some synthetic drugs are proposed for some other diseases, now can be repurposed for some others as well. So this is my strategy to tackle this kind of drugs and their side effects. The diseases and the side effects of these kind of drugs. So multi-drug resistance and side effects are my focus. I aim to uh, find out the real drug targets or the vaccine candidates, and for which I try to find out the indispensable proteins or the biomarkers. So that is my strategy one for target identification. So the other strategy could be for drug repurposing, which is like, for example, I collect sample natural products from different lands and sea products. Sometimes this can be seeds, some fruit, some leaves, and so on. I go for solvent extraction, do some antibacterial assays, get some chromatographic separation of them, go for partial screening, and then do some pharmacokinetics to yield up something which could be of worth. And then there's another drug discovery route through some kind of um, transcriptome analysis, which I'm not going to talk about in this particular case today, because it would be unnecessarily dragging the talk. We'll just focus on the first strategy of mine, and we'll talk about how I have developed the processes and the models. So the target identification is my first strategy, as I was trying to tell you. The process is actually to analyze the network with the features and the first to understand the overall structure, then understand the different centrality measures. Now, I didn't tell about certain more different types of features. I just talked about the global type of centrality measures, which are the different ones over here: degree, then closeness, betweenness, and the prestige centrality or the eigenvector centrality. I just talked about these uh, in the interlude, or rather, you can say prelude, I would say, rather. Sorry. So, in overview, I've just talked about these, which are global centrality measures. Then, there are certain, click centrality measures. A click is wherein a particular protein is connected to all other proteins in their network. So that means they are so tightly connected that you cannot, like, for example, if you just target one such, you disrupt almost all of them. So this is forming a kind of a core. So their density is very high. Remember, I talked about density over here. So there's these kind of local centrality measures are also my focus sometimes, and this is called density of maximum neighborhood centrality, DNMC, maximum click centrality, and so on. I'll talk about them. And then something, as I was trying to tell you about the connectivity, tight connectivity. There are certain kind of connectivity. For example, think tree. Okay, if you just think of a tree in the winter, if the tree sheds all of its leaves, imagine that in the spring it can regain, because the main stem or the main core of the tree is still intact. Now consider this in the case of a network. In the network, there could be certain, peripheral connectivities, these kind of, like branch types, which if you just connect or disconnect these, if you just prune the tree with the leaves, okay, or even the branches, what would happen? Nothing would happen to the tree, because the stem still is intact. That's what happens when the spring comes. The branches, they all again come up to life with the leaves, new leaves, etc., and so on. So you need to understand that in case of certain cases, we can just target the core. If we can target the core, then we really can actually tackle that particular disease. Because if you target the core, then all those kind of proteins which are very important are disrupted, and the particular organism, let's say the bacteria, is done with, means it would die. So that is something we call as core centrality. Okay. Then there's something called functional modularity. This also I have not talked in deep detail, but yes, I can brief over here as well. So what happens is, let's think of different pathways. As I was trying to tell you a little while ago, there are interconnectivities and intra-connectivities of different molecular networks, right? I remember, you remember I showed you a very brief 1.5 minute video. In that, they were trying to also tell you that a particular protein can be important in its own pathway, can also be important across different other pathways. Now, if you think that way, then there would be, if you now map up those within the module degree and their participation across. So participation coefficient tells us the connectivity of that particular protein across different pathways, and within module would tell us the functional modularity of that particular protein within its own pathway. Okay. So if you now actually make up this kind of quadrant of this kind of networks and their connectivities, so there would be a particular place which is having high P, participation coefficient is called P, and within module degree is called Z. So that this high P and high Z is what a particular quadrant wherein certain proteins can lie in here, which means a particular protein could be so crucial that if you target over here, then what would happen is you are disrupting the whole system actually, isn't it? Now, this is very good for a bacterial system because we want to kill the bacteria, right? For those kind of infectious pathogens, right? But then think of cancer. Now, if you target certain kind of proteins over here, what you are doing is you are actually disrupting the particular protein which is connected to so many different other proteins across, as well as it is in its own network. Okay, so then what happens is that this would be a big problem. And in fact, this is the biggest problem that we are facing these days, and I'll talk about that in my model next. Okay. So generally, as I said, I build up networks and I prove them to be real networks. And what are real world networks? Real world networks tells us that there are, like what you call it, kind of a high number of connectivities across very low number of proteins, as well as very high connected proteins are having very low in number. So that means, let's say, think of a social scenario. The very big millionaires actually, they are connected to loads of people, may not be the case, but then they have loads of money. So they have huge money, but they're connected to very less number of people. But then, and they have loads of money. But then consider the downtrodden or even the middle class, they are connected to so many people, but they have very less money. So that is the real world scenario. So we'll see that whatever we do should be obeying a kind of scale-free network. That means it should not be kind of in a falling in a scale, or it should not be directly proportional. As the number of edges or the nodes increases, the number of edges also increases, and so on. So that's what it is. The graph theory comes into play. So we'll talk about these. Okay. And just a minute. So now, let us talk about the different models that I actually have worked upon. I'll just give you examples of two such models. So this is one such, the model for typhoid. Okay. So the causative organism is Salmonella typhimurium or Salmonella typhi. Now, earlier people actually had just related with typhi causing typhoid in humans, but these days typhimurium has also been found to be infecting the humans, and they cause gastrointestinal disorders. Okay, so what they generally do is the salmonella, the organism, they actually inject some kind of secretory proteins into our gut lumen. So this is our host cell membrane, okay, our human being, okay, and this is the bacteria. So bacteria actually puts up a needle and injects some kind of secretory molecules, and these molecules, when they enter in our system, they cause our infection. Okay, whatever infection we can think of. Now, you just think of this scenario. What could be the most important protein? When I started off with working in this kind of salmonella system, by the way, let me tell you that I was not by my PhD like experience. I was not a dry lab worker. I was a complete wet lab worker. So when I went to onto the Indian Institute of Science, remember I told you, so from Bose Institute, when I went out to India for science, I have started off working with salmonella, but that was wet lab work. So when I was just seeing this picture one day, I was just wondering like what happened, like which is the most important, which one I can target. And then what naturally comes up to my mind is that okay, these are the secret proteins. So whatever needle actually pushes them, if we target them, then they cannot push them. But then what happens if there are certain other mechanism which actually still are able to push those secretory proteins into our system, because these are secretory. So uh, which could be the most important ones? Why not actually stop the secretion itself? So there are proteins which are chaperone proteins which actually help the secretory proteins to get into shape. You know that secretory proteins, chaperone proteins actually are those which actually help the packaging of certain other proteins as well. So Salma has such kind of chaperone proteins, and my hunch was that yes, maybe this chaperone protein is the most important one. But till then, people have worked with so many different. This is the scenario: the pathogenicity island, the genes, the this is a genomic island scenario, wherein we call them as Salmonella pathogenicity island 1 and 2, comprising around about, let's say, 40 such kind of proteins. And then SPY 1 and SPY 2, they had so many proteins. And my question was like, which was the most important? No one has the answer those days. So I actually started building up a kind of a model for this. So what I did, I started off with my pathogenicity island database, and then found out that there are 5 such pathogenicity islands. SPY 1, this is SPY SPY 1, then there's a SPY 2 over here, then there's a SPY 3 over there, and then that's SPY 4, and then the SPY 5. So these 5 pathogenicity islands make up the pathogenic proteins for Salmonella. But then across these pathogenicity islands, there are certain proteins, other proteins also, which you can see over here, this in the SPY, around the vicinity of the SPY. Then within the SPY itself, TTR, R, TTR, ACE, and so on. There are SSRB, SSRA, and so on. There are several other such kind of proteins which are signal transduction proteins, and these signal transduction proteins are parts of this kind of spy proteins. That means they also have some kind of roles. So whether they have some role or not, how I should understand, how would I understand that? So for that, what I did is I built up some kind of positive and negative control. So I built up all those spy and made up a network for spy. So this is all spy. A is all spy. All TCS means two component systems, the signal transduction proteins. B is that, just two component system proteins. And my real experimental network is this C, the all spy TCS. Now, what I did is now I analyzed all these with those kind of global centrality measures. Remember, I told you degree centrality between closeness, betweenness, and eigenvector centrality. Now, what I see in B is what? B is all two component system proteins. So even though B shows that ENVZ is a particular protein which is having high, all of these, it says unanimously this very high. So it's a environmental protein which actually senses all the signals from outside, and it has high centrality measures for all across. You can see this. But you see that in the all spice TCS scenario, ENVZ is not within the top 10, which indicates that ENVZ may not be so important. And if you see for all spy TCS, this is the all spy TCS case, and even in that particular only all spy, it is a similar scenario for INVF. This is the invasion protein F. Okay, so this is a similar scenario. So whether or not the two component systems are present there doesn't make any difference. This is being proven by two scenario, as in the INVZ doesn't come in the all spy TCS, and more of that, the INVF stands out as a number one scenario protein for all these two cases, like all spy only and all spy TCS also. Despite TCS being there, INVF, and there are all these kind of spy island proteins only topping up. But then how I can actually consider this to be the unanimously number one, because these are not really having the maximum values for all the cases, okay? And it is not same for all across. For example, SPTPP is almost having a better, like this yellow is closer centrality than INBF. So then I actually had to resort back to the core centrality measures to understand whether these kind of proteins are really within the first, like are they are the real core proteins of that particular bacteria. If I can target the core proteins, maybe I can knock the particular bacteria out of the system and then the disease can be cured. So what I can do is I did this again. A is all spy, B is all TCS, and C is all spy plus TCS. So you see that the innermost core proteins are actually the same and were divided of the two component system proteins. Again, it has been proven that two component system proteins, even though they are signal transduction proteins, doesn't have anything to do with the virulent scenario and has got no role. So then I again actually then move on to the local centrality parametric measures, but still I actually had kept the two component system proteins as well. Okay, so this is again all TCS, and this is all spy plus TCS. And in that, I find that if I do a kind of a maximum click centrality in where all the different disease proteins, or rather the virulent proteins, the pathogenic proteins, are highly connected with each other, in there their neighborhood is also very highly connected. There is a maximum neighborhood centrality, and in their density of maximum neighborhood centrality, that is GMNC, also tells us that okay, they have different close tight pockets of proteins which are connected, and their density is very high actually over there. So in there across, if you do a Venn diagram of all of these kind of measures, we see that CA actually tops up. Okay, so CK is that same protein which I showed up in my model, and that is that particular Sharon protein, CK protein. Remember? So now I got some clue that yes, CK could be the one. And again, as usual, this particular different two common system proteins actually were very much falling out in all those kind of like coming up to be the very important ones. Only that we understand that there can be the chemotactic protein or the flagellar protein, FlyM, CHER and CHEV and so on, even CHEVI also. So these kind of proteins are quite kind of important. We understand that they might be important in the disease scenario, leaving all other two component system proteins. And while it was there in the all spy scenario, leaving all the two component system proteins is just the the other situation wherein we see same CK topping up in this case. So we get an understanding that yes, the CK protein could be an important one. So what I did is I actually did a kind of another gene expression analysis, and from there, what I inferred is very interesting. So what I did is the lab that I was connected to in the at the Indian Institute of Science, Bangalore, India, were walking, and we found out that LACI, not my word though, that LACI needs to be present for a particular bacteria to be non-virulent. Remember this. So if there is no LACI, that is a pathogenic condition. If there is a LACI, there is a non-pathogenic condition. And CHEVI being one of those chemotactic proteins which are helping the bacteria to move into our human gut, if I mutate that, then maybe their movement is restricted. So 4 hour, we have found out that 4th hour is the state wherein this mutation works very nicely. So what we did is we again did that local centrality measure analysis, MNC, MCC, DMNC, and so on. And we found that CK turns out for all spy TCS. I'm not talking of these all spy or all TCS because in any case they would be of no importance. But then for all spy plus TCS, CK is unanimously affected for all of these cases. Okay, so this is the case where it is a minus LACI. What is that? Means it's a pathogenic state. Okay, so in that you'll see that CK is there. This is a CK. Now, if you see a plus LACI condition, which is a non-pathogenic condition, you will see CK is nowhere. You see, there's no CK at all in this case. That means if you do a gene expression level study, and if you go for understanding the sensitivity measures for the local cases, as in like find out which are the important, like virulent proteins which are cross highly connected in the disease scenario, CK is never expressed in the non-pathogenic condition. But you see this in the 4-hour mutant CY condition, CK is unanimously, even though there are other proteins, but CK is the unanimous one, which is actually nicely expressed in this particular state. That means it is actually coming up slowly in this particular state. And when it goes to complete pathogenic state, these are the proteins, these 10 proteins are nicely highly expressed. This is the pathogenic condition minus Lakai. This is the somewhat pathogenic condition, and this is the apathogenic condition or aberrant. So we come to this conclusion, whether through eigenvector, the global sensitivity measures, whether through K-core analysis, the core analysis, or even through the microarray local sensitivity measures, we find that these top 10 proteins, of which CK comes out with the highest value. And so it turns out that CK is the most indispensable proteins along with certain others as well. So we published this paper in the Journal of Theoretical Biology, and you'll be surprised to hear this, that it was published in 2014 those days. And even after 10 years, that the particular articles which comes up to me are all revolving around CK. Only last year I have reviewed an article from Frontiers wherein they had actually seen through several serovars of Salmonella, all having SNPs in CK, which tells me finally that yes, I was correct, but then it was completely unnoticed and my paper was never cited. So I mentioned that why don't the reviewers, why don't the authors actually look into this particular paper which talks about CK being so important since long. So they really understood, and when this was also noticed. So, but then I didn't stop the model typhoid. I went on for bioinformatic analysis. I came to know that there are 17 such kind of salmonella pathogenicity islands. Okay, and we have made all those kind of different islands and their networks. I'm not showing the networks, and then their protein interaction analysis and the Venn diagram for the eigenvector between its closures and like the degree. Now, why I have not done the local one? Because it's the whole genome. So what I've done is I've done the whole spy, and I've done the whole genome actually of all these networks, and I've shown that this is the real network which actually follows the rule that it's not an arbitrary network that we have built up. And if you go for the core, there are 150 such proteins. You see, this is 150 first, that wherein everything collapses. That means if you now need to target Salmonella, maybe there are 150 such proteins across the whole organism of Salmonella which you need to target. How is that possible? Now tell me. So this could be one of the reasons why Salmonella or several such other organisms are developing multidrug resistance. Salmonella is notorious in developing multidrug resistance. Okay. So what we did is we actually started doing the kind of, you remember the functional modularity analysis, wherein we talked about the participation coefficients and the within module degree. And if we see that R7 actually is not going into the details of these terminologies, but then this particular protein actually has a high Z and high P. So this, if you target maybe, or even this high, quite high P and quite high Z, R6 or R7 are those kind of proteins which you really need to target. And you will be surprised that whatever I've seen, no one has worked upon that the 7th quadrant protein putative transposes, when people have started working on slowly on the 6th quadrant protein, plasmid transfer protein or glutamate synthase, the PTS system glucose specific, and these are the particular types of literatures which came up around that time. So you see that this is a TN5 transposes STY0115, this one, which actually helps bacteria to share antibody resistance genes. Can you see this? So this was actually also told in my literature, my analysis, and it came up in 2012. My publication came up in 2014. Around 15, because this was the whole genome. So plasmids are known to be the powerhouse in 2008. Then PTSG, you see PTSG over here in the 6th quadrant, TREB, NAGI. So these are all there. This is TREB, this is NAGI. So this is the uncoupling of phosphotransferase system, GLTB. And so these GLTB is in the 6th quadrant. You can see this Hemi is here, okay? perfluorinated decarboxylase. So these all are important proteins which people really need to target. People have already started working on them, but you see that their approach and my approach are completely different. So if they adopt my approach, it would be much easier for us to quickly understand the scenario and then target the real proteins on. So the recent targets are bacterial acyl transferases, which have been shown in the fourth quadrant. And the fourth quadrant is like within which the high, there's not across this within module. So this value is not high for R4, but participation across different functional modules or rather different pathways are high. So R4 is also quite a good one to target. And really in 2016, exactly the same time when this particular work of mine got published in a book chapter, this came up. So we were just ahead with time, or rather ahead in time, I would say. Now we'll talk of the second model and we'll finish up the work, the talk. So the second model was on cancer, and we actually started off with extracting information from Biocarta, Reactome, Pathway Interaction Database, and Kyoto Encyclopedia of Genes and Genomes. This is nothing but the KIG we are familiar with. Biocarta and PID have actually been discontinued thereafter, and this particular work got suffered many a times. I started off this work in 2010, and then I moved on to Germany, and this particular work, as I was telling you, I was not a completely dry lab worker. It got halted, and then when I actually at some point of time, I didn't tell that, I moved on to the Institute of Mathematical Sciences at Chennai, India, wherein I actually have restarted this work. When I left and went to Malaysia, so then that time there was a huge flood in Chennai and all my data were washed away and the servers, because the servers were shut down. Again, the work had to be restarted and whole work, reworking had to be done and so on. Someone helped me, obviously. So we did this kind of interactome analysis, and this is a whole genome, and we did all those kind of parametric analysis, and we saw that the power law, whether it fits. So this is not a random network, this is a real world network, and we did this kind of analysis also for the whole genome. So we did a kind of random analysis. Let's say if I now don't actually have the real whole actual network and randomly disrupt all these connectivities and connect them randomly, and then try to see if it fits the power law, you see that everything is fine. Power law probably would still fit in, but then the within module to participation coefficient, everything crumbles, that the whole network crumbles. Means our particular model of this particular category, this particular network was the real world network and not just an arbitrary one. So what we did is we actually wanted to actually get some idea on the side effects of the cancer drugs. Okay. So what we did is we actually found out that there were like this kind of drugs which were getting different statuses across the drug banks. For example, they could be withdrawn, they could be illicit, they can be approved, they could be investigational, they can be experimental, and for some they were like status not available, but for some others also there are nutraceuticals, so they are really not drugs, anyone can take them, doesn't need any approval. But then certain others obviously needs approval, and then they are in the stage of investigation and so on. So these kind of clinical phases actually leads a drug to be approved. So there can be without clinical trial like these, and there can be with clinical trial like these. And there are certain drugs which can be illicit or withdrawn after some time. Now, you remember the different quadrants, 1 till 7, which I talked of a little while, just a while ago. You see the 6th and 7th quadrant having the without clinical and with clinical in equal proportion. So whether you target the 7th quadrant or the 6th quadrant proteins for different new drugs, the one case is almost same. Now, if we go down to this investigational, means the with clinical trial, if you know break them down, investigational, approved, illicit, and withdrawn, we see that the 7th quadrant are the most approved ones, and 6 quadrants have even illicit ones as well. Okay. But then if you see there are nothing investigational in the 7th quadrant, that means new drugs are not being developed. So we wanted to know actually whether we really can develop some idea about how new drugs need to be targeted. Okay. Or rather, what are the proteins which really could be targeted for developing new drugs. So now we come back to our same local global measures of betweenness, degree, and eigenvector selectivity. Eigenvector sensitivity. I have not taken closeness because closeness has never been shown to be connected with drug development and so on. So degree is like how much I am connected to others. So even a very high connectivity can make a particular protein very important, or connectivity between several other virulent scenario can make a particular protein important. So betweenness can become important. And eigenvector centrality, like what I said, the most important protein connected to all other most important proteins. So what we did across 7 quadrant, we have done the without clinical, with clinical trials, and their significances were being shown, and we have actually broken them down to different categories of the drug statuses also. We found that the sixth and seventh quadrant had some high illicit and withdrawn also. Okay, if you see this sixth and seventh quadrant, for example, the seventh quadrant has high illicit and high, if you consider the with clinical trial, so in there you see that not just the betweenness or the degree, the eigenvector centrality actually tells you the real picture, because in this you see the significance is not very clear, because you see the kind of a bar, the error bar that shows, but it is very significant in the eigenvector centrality scenario, that you really can understand that yes, the with clinical trial drugs are much better off via the eigenvector centrality measures than via the betweenness centrality measures. Forget about degreenness, because in there you cannot actually see much differences across everything. And in there also the picture is not very clear for betweenness, but in here eigenvector centrality, it's very, very clear. So you can still again understand that eigenvector centrality turns out to be the most important one. And if you see the different other differences across the different measures of the drug statuses, so approved, illicit, withdrawn are kind of very much confusing. So what we did is, sorry. So what we did is actually is the impacts we have found out across the different quadrants with those drug statuses, and we found that only via eigenvector centrality, not via betweenness or degree, that we could really stand out with the in the approval or the illicit, the approval and the withdrawn in the seventh quadrant to be very high. And this is for seventh quadrant, we are not very sure because the error bar very high. But then you see the sixth quadrant, it is nice that there are these investigational and the the approved ones standing out very tall, high for eigenvectors, and the illicit and the withdrawn ones are very low. So this sixth quadrant could be the ones which could be the proteins to be targeted and probably would not cause the side effects. And in fact, we are just around about the end of this talk, so please don't go. The point is that this is the conventional approach, and we found that across other people's literature and their scenario, TP53 is a particular cancer protein which is being targeted earlier, even SRC and so on. But we found out that these kind of proteins which were earlier being targeted, AKT1, TP53, CAM1, June, okay, so these are of no use. For example, if you now target these kind of proteins, what we end up is having side effects of these kind of proteins, because these are very essential across several other pathways also, and it affects the normal cells. So instead, you see my approach of having the 20th core, having the R6 quadrant and the eigenvector centrality as the measure. There's a particular value that we have found out, threshold. We're not going to that. And that actually would tell us that, see, these are the kind of proteins that are very important. And you would be surprised to see that PIK3R1 is the protein which actually was standing out to be the protein exactly the time when this particular paper was getting published. In fact, there was a particular paper in Science which tells us that PIK3R1 is nowadays quite a good target to think of. So we just then proposed, and these are the different pathway analysis of all those quadrants, which shows that R6, the involvement of R6 means that this quadrant has got all different types of pathways getting connected and still standing out to be having nice, less side effects type of proteins for targeting the cancer. So a side effect-free method of identifying cancer targets, drug targets, was my next publication in this line around 2018. And I've later published several other types of models. For example, I talked about salmonella virulence. Then we have identified common proteins across 6 different cancers, then proteins across in diabetes and rheumatoid arthritis. And we've done some kind of these days I'm doing artificial intelligence also as cancer biomarkers. So this was the paper. And then I've done some kind of urinary tract infection proteins also for quorum sensing of Proteus mirabilis, and for Acinetobacter baumannii, which is a very deadly multi-resistant organism, and several other like schizophrenia. Then this is for autism. These are for different shared diseases like autism, diabetes, and rheumatoid arthritis. And this is ankylosing spondylitis that we have done some work. And later on, I would not focus on this part because many of people knows this part. So we've actually done some kind of multilateral resistance protein. We've taken them, and sometime back it was Dr. Shiva's group I have collaborated with, and we found out that this XR770 is one which actually can be targeted, and we've done some kind of computational study. So the second part of the study is that it shows that XR770 indeed is a kind of protein which can be really nice target for kind of this salmonella proteins. And so we have tested several typhoidal drugs and non-typhoidal drugs and several other HSP modulators or inhibitors, because remember I talked about salmonella protein CK. So CK is nothing but a heat shock protein. This HSP is nothing but heat shock protein type. So we have taken several such as HSP inhibitors, and across we have found out that this CK proteins nicely gets docked with XR770, a kind of a particular drug molecule which can be utilized for repurposing these days. Actually, it was like utilized for some other cases, but now thereafter discontinued, and we are now proposing that this can be like targeted. So this is another way of actually understanding that if you have the real target known, CA, for example, other than DNAK and CG in case of salmonella, let's say, for example, and this was the dynamic simulation, we really can actually come up with a very good like drug being developed against that. And so we can actually shift on to repurpose such kind of drugs from phenylalanine moieties to phenylenophyranone types so as to target or combat the multi-resistant salmonella serotype. So I'm not going into the detail of each of them. So there are several such kind of works being done for a kind of a Malaysian fruit-like Nepalium lapicium, and then Gracillary edulis is a kind of Malaysian seaweed, red seaweed found in the seashores of Malaysia, even also found across several other seashores across the world. And so I'm trying to find out some kind of database, and then I really would like to acknowledge for my research the Malaysian government grant, the Indian grant, and